So to begin, one of the most fundamental aspects in the biological thought relates with the understanding of biodiversity on Earth. And to research this, we need to observe the distributional patterns of such biodiversity in the planet. In this heat map, we can see that across different taxonomic groups, there is a pattern where the highest concentration of living species is found in the tropics, and especially in the Neotropics. In mammals, there has been a constant taxonomic effort over time, and therefore it is a relatively well-studied taxonomic group. However, its diversity has not been completely described, and we hope that in the next 70 years, approximately 3,000 new species will be discovered. Of the described species, there is estimate that there are 6,400 mammal species, with the most species richness inhabiting the Neotropics. However, we face significant challenges in identifying even the known diversity. We particularly have problems identifying species with complex morphological diagnosis within small body size lineages. There is an example of those characters, and this diagnosis is performed, or the diagnosis for identify the species is performed by an expert taxonomist, and this needs the sacrifice of the organisms. And for example, these two opossum species of the genus Marmosops can only be identified based on the lateral expansion of the nasal bone in the maxillofrontal suture, and this requires an expert taxonomist and to sacrifice these organisms. DNA sequences have been implemented to overcome the morphological identification issues, but however, traditionally, PCR-based methods require specific primers that are not always available for non-model organisms, and almost all neotropical cryptic biodiversity is composed by non-model organisms. As well, this methodology needs large facilities with robust equipment, and this can be time and money consuming. Traditionally, specific mitochondrial genes have been used to identify species, but these mitochondrial genes have changed over time and are widely and are very specific for different biotic groups. And this has generated that the databases or the reference databases are incomplete and heterogeneous, and not all species are represented. So we may find ourselves with the following scenario. Suppose that we want to identify this marsupial species by using mitochondrial DNA sequences and comparing them with a reference genomic database. But the markers that I managed to amplify are not available for these species in the reference-based databases, or there is no available genes for the marker that I managed to amplify. So one possible solution to this is to sequence the entire mitochondrial genomes to maximize the probability of identifying these species. Oxford Nanopore makes it possible to sequence the entire mitochondrial genome without the need and avoiding the problems of PCR-based sequencing methods. Additionally, with the MinION device, we can obtain and analyze sequencing data even under field conditions. This study has been developed by Juan Diaz, the PI of the research group where I belong, and we are testing this portable sequencing technology and the fungal flow cells for mammal species identification and description. Our goal is the approach of development of a new approach for the rapid and accurate species identification in situ based on whole mitogenome sequencing using the MinION sequencer portable device. And how we are doing this? So we conducted fieldwork in 5 different locations in one of the world's most biodiverse regions, the northern Colombian Andes, and we sampled small mammals, including bats, rodents, and opossums, by taking tissue samples that did not require the sacrifice of the individuals and extracting DNA from those tissues. Then we perform shotgun sequencing and the following computational workflow for base calling and quality filtering. And I will explain later how species identification works at this point. Then, to obtain the mitochondrial genomes, we implemented 2 strategies using whole genome sequencing data and mitochondrial filtered reads. For the whole genome sequencing data, we applied de novo and reference-based assembly using fly and revealer respectively. Then for the mitochondrial filtered reads, we use Minimap2 program to filter those reads, and empty blaster, which is a script. And we applied the same methodology, assembling both de novo with fly and reference-based with revealer. And lastly, we annotated the mitogenomes of the 6 resulting pipelines. We found that with the MinION device and the fungal flow cells, you can generate enough data to assemble mitogenomes. And the sequencing yield was better explained by the ports available for immediate sequencing than by DNA concentration or quality. In this table, we see the percentage of genomes assembled by each assembler for each data type, and we can given 2 sequencing yield range. And we can see that fly assemble most mitogenomes if the datasets has equal or greater sequencing yield than 550 megabase pairs. But this assembler was poor at reconstructing genomes from datasets that has lower sequencing yield. In contrast, revealer was very effective at assembling mitogenomes even when the datasets has moderate sequencing yield range. We also found that although fly needs better sequencing yield range to assemble, these de novo assemblies tend to have a slightly better quality than those assemblies assembled with revealer. These annotated and mitogenomes these assembled mitogenomes enable us to obtain a reliable species identification. However, it requires prior taxonomic knowledge of phylogenetic systematics, for example, and this can be time-consuming and needs the expertise of a taxonomist. Therefore, we develop a second pipeline for species identification from the base called data to make a rapid species identification without the need to be an expert. These analyses consist on mapping those mitochondrial reads to a mitochondrial DNA reference database, simulating 3 different scenarios regarding the amount of information available for the sample species. When no information is available, the complete metazoan DNA reference database, when sample identity is known at order level, or when sample identity is known at family level. This identification pipeline enables us to identify the species in 2 hours or even in a few minutes when the sample identity is known at order or family level. And these methods enable us to obtain 21 mitochondrial genomes belonging to non-model organisms or neotropical small mammal species, which 2 are endemic to Colombia and 4 are new genomes to science. Oxford Nanopore makes it possible to make species identification without the need to sacrifice the individuals, to do so in a matter of hours or minutes, and even under field conditions. And this is extremely important in scenarios of conservation concern. And also, it is contributing enormously to filling the gap between the biodiversity we know and the biodiversity that remains to be discovered. And by enabling researchers to identify the species rapid and accurately, Oxford Nanopore may revolutionize the field of biodiversity research, especially in institutions with limited budgets such as universities or governmental entities in developing countries. I'd like to special thanks the Universidad de Afit for funding this project and its computational center, Apollo, for supporting us in the development of this research. And I want to give a special thanks to Semillero de Biodiversidad for this hard work and committed. And I want to thank you all for hearing this.